Welcome to part 2 of our histology drawings. In this part, we are going to be looking at the cochlea histology and the nasal epithelial histology. Now, if you remember from lecture, the cochlea is actually the spiral organ. And you have it starting at the oval window right here. And it goes, and basically like a snail, it spirals and spirals until it reaches the apex, where you have a second tube that's going to spiral back down and exit in a structure called the round window. So the cochlea is actually this spiral-shaped structure. The first histology slide I'm going to show you, they took the cochlea and they cut it down like that. So they did a cross-section. So you're basically seeing a cross-section of this tube. So here we're basically seeing a cross-section. We're going to see this repeated structure, a scala of vestibuli, a cochlear duct, and a scala tympani. So here's my picture from the lecture. So we have the stapes pushing on the oval window, and there's this long continuous tube going to the apex, and it then goes back around, and there is a structure in the middle here. So that picture on the last page is basically a cross-section where somebody has gone like this, and you're looking at it. Oops. Let's put the arms in the right place, like that. So that is what this histology slide actually is. So here's that cross-section that we're looking at. And we have the repeated tubes. Here's the top tube, here's the bottom tube, and this is the two tubes that they're forming. And we have the cochlear nerve here, and we have the spinal ganglia. So this membrane right here is going to have hair cells, and the hair cells lead to the spinal ganglia, which is a well-named ganglia, which is then going to go to the cochlear nerve. So we're just going to focus on one of these structures when we do our histology drawing. So here I zoomed in. We have our two tubes, our tube scala. We have our cochlear duct in the middle. Here are the hair cells, and there's the neurons attaching to the spiral ganglia, and then the cochlear nerve is off the screen. So let us begin. So the whole cross-section is going to be a lopsided oval. And there's two membranes. There's going to be one membrane there, and that's the thicker membrane, and a thinner membrane like this. These membranes are the vestibular membrane and the basilar membrane. So draw your membranes and label them. Basilar membrane is the thicker one on the bottom. Vestibular is the thinner one. And on the real histology slides, they're actually usually broken. Now, the basilar membrane is the base of the hair cells. So we need to add some hair cells. So hair cells are these little, tiny, almost cuboidal cells. They're not quite cuboidal. And there's a couple different types. There's some all by themselves like there. There's some hair on them. And then there's going to be a big structure in this middle here. So here are our two sets of hair cells. Now the hair cells have a membrane on top of them called the tectorial membrane. And usually you can't really see it in the microscope, so I'm not going to add it onto here. But again, I recognize this as the basilar membrane because it has hair cells sitting on it. And it's usually thicker, and this is the vestibular membrane. Now, there's neurons, or axons, coming from the hair cells, and they are going to be going out to the spiral ganglia. So let's draw some axons. And since this is a bunch of axons, it's a bundle of axons, in fact, that's a nerve, and it's going to the spiral ganglia over here. Now, we have three cavities formed by our basilar membrane and our vestibular membrane. These two get the name scala, and they actually form a duct in between them. Now, since this tube is formed by the vestibular membrane, it is called the scala vestibuli. This one down here is the scala tympani. Now, the hair cells are located in a duct, and this is the cochlear duct because it's the duct in the cochlea. So take a minute to make your drawing and to label everything. On the lab exam, you'll get a picture like this. And this is sort of how your picture hopefully ended up. We have two membranes. We have the thicker basilar membrane here, and the thinner vestibular membrane on top. Now, the vestibular membrane is going to form the scala vestibuli, and I will circle that in green. And it probably goes like that. And the basilar membrane is going to form the top of the scala tympani. Again, I have no idea why they call it the scala tympani. These two membranes come together. They're going to form the cochlear duct in the middle. Now, on the scala, on the basilar membrane, there's hair cells. And you might be able to see these little fine hairs right there. The hair cell really actually looks like this. It has these stereocilia. That one's not a stereocilia. On the back, and they're sort of all connected to each other. So that's what the hair cell really looks like, and let's add a nucleus to it. So, expected to have to identify something from this slide on the lab exam, and here's the spiral ganglia over here. So, our last one is going to be the nasal epithelial slide. And this is important because that's our sense of smell. So if you remember from the previous lab, we have our olfactory nerve coming along. We have the olfactory bulb right there. And there's extensions into the nasal cavity. And these are all bipolar cells in there. So this picture is actually for a future lab, but it shows what the epithelium looks like under low power. So we have all of these bipolar cells right here. And they're going to actually have extensions called their dendrites that are going to go, and they're going to go into the epithelial. Now, because this is pseudostratified epithelial, it's really hard to see, oh, that's actually the dendrites from here, not a cilia. So all these bipolar cells. And then their axons are going to actually form small bundles, and they're going to go out and go through the cerebriform plate of the ethmoid bone. Here's the ethmoid bone. And into the ganglia of the olfactory bulb. So this is the slide you would usually draw. We have our epithelial layer here. We have our epithelial layer here. We have our bipolar cell layer. And then we have just the submucosal. You can't really see the nerves that they're bundling into. So this is what I want you to draw. We are going to have our epithelial layer right here. Just draw some stratified squamosal cells. Ooh, let's do a goblet cell. I love goblet cells. That's a humongous goblet cell. Some more stratified squamosal epithelial. Let's do another goblet cell. That's a good place for a goblet cell. And a little bit more. And do you know what we're going to label this? Pseudostratified epithelial. Now I want you to take a different color. Now, it would be easy just to take the dot clone tool and make a bunch of black dots. Yes, I am thinking about it. But let's draw a couple nice bipolar cells. And they're going to have their dendrites going through. So let's draw some nice bipolar. And let's draw the axons. They eventually go together. And label these bipolar cells. Now, since this layer is so thick, I think you need some stress management. Take your black pen, preferably a large diameter fluffy pen, and then make nuclei. Now don't kill you. Let's do some nuclei. And that's our bipolar cell layer. That's all this slide is. It's really exciting, isn't it? But I really want you to understand we have thousands upon thousands of these bipolar cells, and the receptors are literally on their dendrites, and it's sticking into the nasal epithelial here, and then it becomes a nerve. So on the last page of the follow-up, this is how I would ask this on the test. I might ask you what organ this is and what the structure is. So this organ would be a cochlea. And then here's the scala basale. Here's the basilar membrane. Here is the vestibular membrane, scala tympani, scala vestibuli, cochlear duct, and hair cells. Oops. So for this picture, I might ask you what organ. It would be a retina. I might be nice and let you say an eye. And we have our sclera back here, which isn't labeled. This is the choroid layer. This is our rods and cone layer. This is our bipolar layer. This is our ganglia layer. So I always either look for the choroid or the layer that has the most nuclei on it, because this one has the most nuclei. That has to be the rod and cone layer. Then the next one's bipolar. Then ganglia cell layer. There's only going to be a few ganglia in most of them. Then this is what a taste bud looks like. So my picture really did look like this. So we have our pore there. Even on this magnification, which I think is 2400, you can't really see the hairs coming out. We have our gustatory cells here, and then we have our basilar cells. So if I go to the other side, this one looks like it got a little squished. There's the pore. Here are the gustatory cells, and these would be basilar cells down here.